Faridabad, India, where she leads the development of Indian Crop and Animal Phenome Databases. She has contributed to the development of FAIR compliant metadata standards and also very active with the STEM outreach. Sonia currently serves as one of the executive committee, member of the International Society for Biocuration. I welcome Sonia to AgBioData community and invite her to present the interesting work her team is pursuing in India.
Thank you, Dr. Sushma, for your kind words, and greetings to everyone. It's a pleasure to be here with the wonderful and diverse AgBioData community, and thank you for providing us the opportunity to present our work, which we are doing at Indian Biological Data Center. So we all know that agriculture stands at the intersection of urgent challenges, that is climate change, food security, and biodiversity loss. While we all witness that genomics has advanced rapidly, and we often say that genomics tells us what could happen, but phenomics tells us what actually is happens in the field. So that's where ICPD, or the Indian Crop Phenome Database, comes into the picture, and it's a national platform for the sustainable archival management and sharing of crop data. So before going into the details about the crop phenome database, I would like to share my motivation behind being the part of AgBioData. In Biocuration 2025, I met these wonderful people, Dr. Leonard, Dr. Sushma, and Dr. Marcella. They motivated me to join AgBioData, and indeed, it's a very enriching experience till now to be being the part of this diverse community. So coming to the Indian Biological Data Center, where we are in India, it's a very new repository, and Indian Biological Data Center is situated at the Regional Center for Biotechnology in Faridabad, India. And it's the National Life Science Data Repository in India. Just a minute, I just have... It's the first national life science data repository in India, and it is supported by the Government of India through the grant from Department of Biotechnology. And it basically enables the implementation of the very first guidelines that were there or implemented for the sustainable reuse of the biological data, that is the Biotech Pride guidelines. And it's an initiative which is a joint project of Regional Center for Biotechnology, National Institute of Immunology, and the International Center for Genetic Engineering and Biotechnology, and National Informatics Center. So we have a disaster management site at NIC Bhubaneswar in India. So this is the location where the Indian Biological Data Center is located. We are located in the neighborhood of two very eminent institutions of repute in India, the Translational Health Sciences and Technology Institute and the Regional Center for Biotechnology. IBDC is very recently dedicated to the nation by our Honorable Minister of Science, Dr. Jitendra Singh, on November 2022. So since then, we are moving with a vision that IBDC will provide data solutions to sustainably archive the life science data that is generated in India, and contribute towards the curation, dissipation, and also to the outreach of the fair data practices within the Indian community, and also engage with the international institutions for possible collaborations and moving towards the fair data and open science. So all this vision is to advance the faster diagnostic, agricultural innovation, better livestock yield, and personalized therapeutics. So basically, what all activities we do here is our first aim is to provide the biological data resources for the sustainable archival of the biological data. We also provide the high computational support to the research community, and we also actively support the project of national importance, and we are also performing big data analytics for in-house projects and also as support to the research community. We generate a lot of user resources so that they are guided towards the better practices and the SOPs towards the submission of different types of data. And we often do and regularly do trainings about the data submission and data analysis. So since the IBDC came into the picture, we have 7 active databases that are presently welcoming different types of submissions. The first portal that we have launched is the Nucleotide Data Archive, and we have another version of this Indian Nucleotide Data Archive under the controlled access. Then we have Indian Crop Phenome Database for the crop phenomics data. We have Indian Structural Data Archive, which is for the biological macromolecules with the functional annotation and different types of structure data associated with all the biological entities. Then we have the database for the Indian Metabolome Data Archive. We also have the Proteome Data Bank, and we also have the Indian Biological Images Archive. Apart from these data types, IBDC recently launched the BioNode, which is basically a multi-omics toolbox. It has 3 different modules where the research students can perform the data analytics based on the structural biology, based on genomics and biomanufacturing. Then, as a part of the national support to different national projects, IBDC is home to the INSACOG data, which is the consortium for COVID, and Genome India, which is the consortium for the 10,000 human genome sample that are recently launched. We also support different projects specific to different mission mode projects of different crops like triticum, oryza, linum, sizer, Carthimus, and sesamum. So as of now, we actively submissions are there in IBDC, and we surpassed the 1,000 TB data, and more than 1,000 active users are there which are submitting data to different portals. As the map suggests that our users are in pan India, and we are receiving submissions from different regions and different institutions that are present in India. So that is a testament, or we are witnessing that IBDC is playing or started to play an important role in the research infrastructure of India. So coming to the what kind of diversity that we have, and we are working on different biological databases from genes, genomes to variation. We have INDA, INDA-CA. For functional genomics, we are working on Indian Array Data Archive. For proteomics, we have this Proteome Data Bank, and we are also working on different databases of phenotype, like ICPD is already there in the picture, but we are now expanding Indian Crop Phenome Database to animal phenome, human phenome, and for microorganisms. And we are also building GWAS repositories with a view that in future we will definitely connect with the different types of different domains of data together to contribute to the knowledge generation. So coming to the phenotype data, as we all know that crop phenotype data is diverse and it often generated in labs, in smaller experiments, to fields, medium-sized experiments, or in semi-controlled phenotyping. Then we have now advanced to the high-throughput phenotyping facilities. But the one thing which is very challenging with this type of data, which is unlikely with the other type of genomics and multiomics data, is that this data that is generated is very heterogeneous in nature. It differ in environments, it differ in the how the data is collected, what all measurements were there, what all scales were there, different devices were being used, different sensors are involved, different measurements are involved, and all these make this data to be very complicated to be in a cycle of fair archival. And then we have different tissue, different stages, different scales, varied names and measurement convention. Then everybody is using different terminologies, and rarely it is documented in a proper data formats, because one reason could be the dedicated repositories for this type of data, and usually it is neglected in laboratories. And as I consider the case here in India, this type of data is very judicially, manually, and more often it is manually collected with very intensive efforts of required from the students to the field workers to everyone, and then it is never documented in a proper format so that it can be reused for the future research, to support future research and knowledge building. So with these drawbacks, this makes this type of a data to very difficult to reuse, because it's never in the process of the fair data cycle. And after analysis, people go on publish the results, and it's very rare that they submit to some public repositories. Or if someone submits, it's generally in Zenodo, which is the journalized repositories, or maybe present in the figures in the papers. And uh, very uh, uh, very often it is just forgetted, maybe as the time passes, as the people retires from their labs. So it's never in the process of a fair data cycle, and it's rarely reused for different purposes. So with this view that we know that as this graph shows that the status of India, as you can see from this graph, is that in India, majorly the phenotyping is done using the traditional phenotyping methods, and we have only 3 facilities in India for the high throughput phenotyping. So this makes that the data coming from the phenotyping studies or experiments would be manually generated, and with lots of efforts, and very few only recently that the people are shifting to the high throughput phenotyping facilities, and that data is automated. So while designing ICPD, we have to keep in mind that uh, the phenotyping data can be of diverse types, from simple experiments to high throughput experiments. And we need a data format which is simple enough, because it's important to motivate the researchers to come to the portal and submit, and within a very small amount of time dedicated from him, so that he's able to get into the habit of submitting and properly managing the studies that are generated from this type of a data. And over the time, when people get into the habit of the fair management of the phenome data, we will be benefited from the collection. So, more specifically, why this data holds very important place for India, because when we consider the case of India, majority of our workforce till now is also employed in agriculture. Around 45 0.5% of the Indian total workforce is employed in agriculture. So it provides a major livelihood support to India. It also adds to the GDP, overall GDP of the country. And in terms of food security, the growth sector, and the budgetary allocations, agriculture is the major area which India needs to focus for the future research, and also to tackle the climate change and different national problems. So this phenotype data is important because this data alone, and also with integration of GWAS, QTLs, and other type of omics data, could be the answer to food security and engineering high yielding and climate resilient crops, selection of varieties based on different traits, stress response, and geographical location. To support precision agriculture, to make data-driven decisions in making the precision agriculture work, and also to compare different types of data and bridging the gap between the genotype and the phenotype data. So what most important here is that data alone is not sufficient. We need to document this data in a way that it could lead to the accurate identification of this data. So there comes the metadata format, which is very critical to make the format in a way that all the necessary information about the data can be recorded. So keeping this in mind, metadata is again, we should be able to document what was measured, when was measured, where, how, and why, and how to connect different phenotypic traits with the actual experiments, environments, methods, and to make it complete and make it trustworthy. It again is important to make the data fair so that it can become findable, accessible, interoperable, and reusable. And it again will leads to the reproduction of the work, validate the findings, and to compare different studies between different seasons. Then integrate this type of data with different environment and genomic data, which again make the data sustainably. So with this consideration in mind, we have developed Indian Crop Phenome Database at IBDC, and the vision is to make a resource which is user friendly, central to our national infrastructure, to collect, annotate, and integrate the phenotype information in alignment with the FAIR principles. And also now we are working to integrate with the care principles and ensure that these standardization, accessibility, and interoperability of this type of data is utilized with proper metadata fields, ontologies, and again to enhance its findability, interoperability, and reusability to empower the research and development. So whenever a user submit data to Indian Crop Phenome Database, we provide persistent unique IBDC accessions, and then this portal can be used for submission of the crop phenotype data. We will sustainably store it, and any user can access based on the access mode of. So currently we are offering open and managed access modes for the submissions. We are enabling ontology based submissions and annotations of the data. Then with a view that we can able to make the process fair, and we will able to integrate data in a manner so that it can be used in future research. So we collect all the data from which is required as from the user, germplasm information, environmental data, experimental design, geolocation data, and contributor and other important metadata. So the inspiration behind ICPD is again the international accepted formats, that is the minimum information about the plant phenotyping experiment and GNPIS. But the ICPD format is not exactly the replicate of MyAPI. It is inspired from both of these, and we have tried to make the ICPD format simple enough so that the research community can easily integrate into their research practice. This is the model is such that any individual consortium can able to document the phenotype data or the phenome data, whether it's a single project, it's a consortium project, it's whether a user has multiple projects, whether a user has single projects. A single project can have multiple studies. Again, a single project can have different crops, multiple crops. So that in a way that data is submitted, managed properly, and we provide accessions and data is accessed. So in this way, any type of phenotypic data, whether it's a simple phenotype experiments involving a few crops or varieties, to advanced field experiments where we have thousands of cultivars, whether involving a single treatment or no treatment or multiple treatments, or it's a multi-year data, this format can able to integrate all of that. And uh, we have three levels. We user can create a project. We will provide you the project accessions.
Recording in progress.
Then we have user have the another level is the study level. One project can have n number of studies, and each study will have a data file. Again, a single data file have many observations. Then the process is such that we incorporate the ontologies from crop ontology and plantium. So this is the overall structure of the Indian Crop Phenome Database, which again tells us that we take organism at the level of project, and organism can be many. We connect to the NCBI taxonomy. Then at the level of study, as one project can have many studies. In study, we can collect information regarding the meta trait. A meta trait is a higher trait, which, for example, if somebody wanted to screen for drought resistance or drought tolerance or salinity tolerance, then he has to measure several traits, or you can say variables, through in that study. So the schema is that one project can have many studies, one study can have multiple traits, multiple author, multiple treatments, a single environmental condition. And then we also provide that user can submit all the associated data of the study to in the same form. Then we collect the genotypes information, genotypes or the very different varieties, the list of varieties. This list can be 1,000, 2,000. There is no limit to that. And then we collect the crop passport data in the form of MCDP. We have these data files which contain the observation units, and we try to again curate the characterization data of the genotype submitted by the author from different gene banks like GeneSys, ICRISAT, and NBPGR gene bank, with a view that we are able to provide user with all the necessary information about the genotypes. And in near future, if we want, any user want to find that I want genotypes with such and such trait, or I want drought tolerant cultivar or any other requirement, he can able to do from this database. So this is the details of whatever fields we are collecting in each section. So basically the Pheno project, we collect the project, which is the highest entity in our model, which requires the title, grant number, funding agency, project description, associated publications, release date, collaboration, and different crops. So in study, each study is again connected to a single crop where we require the information regarding the title, description, start and end date of the study, location, experimental design, growth facility, what all tolerant and susceptible cultivars they are using, what environmental and growth conditions are using, whether the experiment is conducted in greenhouse, field, etc., what light, temperature, and other conditions are required or used in the study, what all treatments are given, what's the treatment agent, description, duration, at what developmental stage the treatment has been given. And then comes the important section where we collect information about the trait which are recorded in that study, which consists of variable unit like trait name, observation, developmental stage, plant tissue, and the method details. Again, lastly, we collect the names of all the authors who contributed to the study. And then we have a template that is generated and which has autofilled these metadata, and user need to provide only the observations. So in this way, template based system, it can be used for recording the observation, and then in the same template you can directly submit to the ICPD. And in the associated data section, he can submit the passport data, he can submit the soil and weather data, and any other supporting data required to completely describe that study. So this is the overall access flow of Indian Crop Phenome database. User need to register to the portal and follow these simple steps, where the step 1 is to register a Pheno project, and then he can register many studies within a project, generate the template, populate the observation data in the template, and submit it to upload it to the ICPD database. Then when any user can access the ICPD based on the data access mode selected by the submitter. He can download the data, he can download the metadata and use in their research. So the template that is generated is something like this. As you can see that it has a section which is auto-filled, and it has a section which contain observations. So when the author generate the template, this template would have all the metadata filled, and user need to provide these observations. And here B stands for the biological replicate and T stands for the technical replicate. This template can be used for documenting the data prior to the experiment, and after the experiment, after the complete documentation, user can upload the data. So in this way, user can manage the phenotype data of the lab and different projects in a very efficient and easy way. So this is the user dashboard, which provide different functionality for the user, where he can manage all the phenotype projects, has functionality to submit new data. It will tell you the data stats. You can download all the files from here. You can see the metadata of the studies. You can see the metadata of the projects. And you can also able to download the details of all the projects. And we will provide you the complete SOP of the submission process. It will also give you one section of browse ontology, because when you are submitting data, it's possible that the ontologies that we are using from crop ontology and plantiome, it's possible that the ontology of your choice might not present in our database. So we have provided this tool where author can browse and check whether the ontology of his choice is there in our database or not. If it is not present, we provide you a form. Author instantly create ontologies and proceed it to the submission. Later, these ontologies are these ontologies are assigned a temporary accession, and later we will submit to crop ontology, and so that it can also contribute to the ontology generation of different crops. So this is so at present, these are the current statistics. We have around 74 users of 41 institutions, and we have around 367 studies with 325 data files, which these again comprise of around 38 lakh or so observations. And as you can see that now ICPD is being used from different states, institutes from different states, and these are the crop which from of the phenotype data from these many crops are available, and these are the data growth over the time of ICPD. So slowly and steadily we are gaining momentum, and we are able to convince the research community so that they can able to start the journey of making their phenotype data fair through ICPD. Although it's very tough because unlike of the genomics data that which is currently is in habit that you have to submit it to some public repositories, phenotype data is very hardly or is in the habit of submitting somewhere else. So it's a continuous efforts from our side also to connect with the research community or the scientists who are doing the agriculture work or are in the crop improvement or research or plant research, so that they can able to document and document each and every type of data, whether it's a transgenic screening, whether it's cultivar screening for different uh, traits, etc. So it's a continuous efforts from our side also to motivate the researchers, and now we are able to get them to the ICPD and trust ICPD for submission of their data. So currently we are having highest number of data from the wheat and chickpea, and we are able to document around more than 300 traits are submitted in the ICPD through different studies. Currently, majority of the data is under managed access, that it is released after some time, after an as the embargo has will be embargo will be reached. Then we have data from different perspective, from abiotic studies, from biotic, and even from the control experiments. So as of now, 37 meta traits are submitted in ICPD. This is a picture which shows the genotype count of the submitted studies, and each study will have the number of very high number of genotypes greater than 15,000 or so, and these are the number of different samples. It includes the biological replicates, so that is why the peaks are so higher. And very importantly, ICPD is becoming the data hub for major mission mode programs on the characterization of genetic resources supported by Department of Biotechnology, and these are the projects which are consortium-based projects from ICAR, ICRISAT, and funding from Department of Biotechnology, and involving many institutions of agricultural research. So these mission mode projects, we have submitted data to ICPD, and now to support their mission and vision, and to make them to create an outreach for their projects, we are creating mission mode specific dashboard at ICPD site, and this is one of the dashboard that we have created for the mission mode project of chickpea, which is the chickpea atlas for national agri innovation, that is called chana. you can say chana is also the common Hindi name for the chickpea. So this is again a project of multi-institution project, and it has thousands of cultivar that they have screened. All the data files are there, which will be released as the embargo passes. And then we have presented the accession details. We have curated the passport information from ICRISAT and other resources. We have collected the characterization information of all the cultivars from ICRISAT, and the pictures are provided by the group. So in this way, we are able to collect the details about the genotypes, about their characterization, about the images, and we are also associating with the other different type of data that is submitted in different repositories, so that this page will work as a common page which will connect all the data related to this project in a common place. So as I'm talking about that, we are using ontologies, and we are providing the services for the user so that it can create ontology also. So we will provide that user can create ontologies for meta trait, stage, plant tissue, and methods. And as of now, these are the stats that these blue numbers are the new ontologies that has created. So for example, like crop ontology, even crop ontology didn't have the ontology terms for sunflower, but here our group from Indian community, they have created the ontologies for sunflower. So the vision here is when the user create or contribute towards the ontology building, we will definitely uh, pursue it, and we will submit it to the crop ontology so that it can get the valid accessions. So as of now, we are giving the temporary accessions to just separate out from the other ontology terms. So these are the browse page. As you can see that you can see the see and also download the projects related to the search. And these are the metadata formats. Any user will have the all the metadata regarding to that study. It will give you how many genotype samples, what's the size, what all authors, what all traits are studied, what treatment, what are the growth and environmental conditions, what are the metadata of project and study. So this all can be downloaded. So at last, we are now working on different aspects of ICPD to how to evolve it to or contribute our efforts to make it a database which can be used by the research community of the agriculture research community. We are working on towards the interoperability with the international phenomics and genomics databases. We are working on ontology curation and its submission. We are now started curating the published data, and so making another type of option for our curators to contribute towards the data submission, because when we write to the researchers, is it's many in the cases they never respond to our invitation to submit. So that is why we are now shifting to the curated mode, so that we can curate publications which have the phenotype data in the same format as of ICPD. So we are able to now two types of submission, which is the original submission submitted by the authors, and now the curated studies that the ICPD curators will perform. Now we also working to provide a decision support system to the breeders and policymakers, so that we can able to help them into their research and their objectives. We will definitely in future will jump into the AI ML driven phenome discovery, and we are actively involved now towards the data analysis pipeline and visualization tool. So how we are able to make this data interactive and make available for the user to do analysis and extract all the data as per the need of the user, whether he wants only some cultivars, some traits through this databases. And to in the broader sense, we will definitely try to integrate this data with the other data types so that we are able to build resources in a crop specific manner, and again to build a useful resource for the agriculture research community. As I'm telling in the beginning that now we are expanding the phenome to the phenome repository. Now this Indian Phenome repository will have four branches, for one for human phenome, animal phenome, and for microorganisms. The work is under development, and very coming future you will see that these different domains are also got integrated into IPR, so that now the animal phenotype data and human phenotype data and microbes data can also be integrated in a way that it can become fair. So this is about the Indian Crop Phenome Database, and these are the people behind the development of the Indian Crop Phenome Database, and also the IPR team. We are all women's team, and we have in IPD we have Mayuri, who is our software developer. Isha and Nivedita are data curators, and for Indian Phenome Repository we have Vibha as the developer. So we work together, and initially the guidance is also provided by Dr. Saurabh Raghuvanshi, Pratyush, and Abhay. So these are the team of IBDC, and Professor Arvind Sahu, Dr. Debashish Mohanty, Dr. Dinesh Gupta, and Professor Nair, and Dr. Shashank Shekhar, who recently joined as head IBDC, are there to guide us. And this is the complete IBDC team. And in future, we keep on expanding. And with this, I think I thank you all for your patient listening, and I'm happy to answer your queries.